Hello, everyone. This is Kathy Go, and I'm here to talk to you about cellular adaptation, cell injury, aging, and death. So that covers a pretty big gamut. So let's begin with reviewing a few key concepts. Some concepts include the functions of the cell membrane, which is acts as a mechanical barrier. You remember this glorious bilipid layer that has so many roles. It really holds the cell together and it allows the cell to control its environment. It's selectively permeable, which controls entry and exit from the cell. It has an electronic gradient, which is able to maintain an electronic gradient, which is needed for depolarization, and that's important particularly with nerve and muscle cells. It plays a role in communication from cell to cell recognition, and interaction, and that's really important in all kinds of levels, but particularly in the immune response and the inflammatory response. Cell signaling is another function that the cell membrane performs. The proteins that are in the cell membrane interact with specific chemicals to relay messages to the inside of the cell. It's very sophisticated and very cool. And this image, you can see some of the transport proteins, those purple cylindrical looking things. That's what those are. And the outer layer is made primarily of cholesterol. And there are little carbohydrate chains that stick out of it. It's pretty amazing. So how do things get through the membrane? This is just a quick review. So as you probably recall, there's passive transport, which requires no energy. And the types of transport are simple diffusion. Things that can move through the membrane that way are fats, oxygen, and carbon dioxide. Facilitated diffusion, glucose and some ions move through the cell membrane that way, and osmosis, which mainly is the movement of water. Active transport requires ATP. There's primary active transport, and that the sodium-potassium pump, if you remember that, is an example of that. And there's also secondary active transport that uses other mechanisms besides ATP to move materials from the outside to the inside. Things that move that way are things like glucose and amino acids. And then there's vesicular transport, and that is one example of that is phagocytosis, and white blood cells use that quite a bit. Pinocytosis is also, that's the little inclusions that happen where they suck in materials through the cell membrane, and a nutrient absorption is an example of that. There's receptor-mediated vesicular transport, and insulin, cholesterol, and enzymes are moved that way. And exocytosis, and that's where things are spit out of the cell, things like hormones and neurotransmitters. So, just a review question. Which one of these describes active mediated transport? Movement driven by diffusion, expenditure of metabolic energy, moving of water down a concentration gradient, or the mechanical force of water pushing against cellular membranes. So which one do you think? So another important feature of the cell, which plays into the discussion today, is cell division. Certain types of cellular adaptation rely on increased rates of division, and the factors that influence that are protein growth factors, genetic factors, and epigenetic factors, which mean things in the environment. It must be strictly controlled. And there are several ways to control the cell cycle, and those include mitogens and extracellular stimulus. So cells communicate with one another through three main mechanisms. There's gap junctions, which connect to where cells connect to adjoining cells and allow movement of small molecules through these tight junctions. There's direct cell-to-cell communication, and that is where mediators are passed back and forth. That's a big piece of the immune response, which you'll hear more about later. And then there's chemical messengers, which are secreted by one cell and trigger a response in another cell. So those are the main mechanisms. So let's get down to the objectives. Objective 1, compare homeostasis versus adaptation. So homeostasis, as review, is that state where there's a maintenance of balance. And the cellular level, it's a normal, adaptive, steady state. Cells maintain a stable intracellular environment using this plasma membrane as one of the features that control that. Cells are able to control cell content, such as water and products of metabolism. Cellular adaptation refers to changes made by a cell in response to environmental stressor. Cellular adaptation is also known as allostasis. Allostasis is the process of achieving stability through that adaptation, usually short term. Stressors include hypoxia, so things that might bring this stress response about are hypoxia, free radicals, intentional or unintentional injury, immunological injury, infectious and inflammatory injuries. So these adaptations can be physiologic, which means beneficial, or pathologic, which means injurious. So here is an image of this process, sort of the sequence. So cells have a couple of choices when stress is applied. They can draw on their reserves and increase functional demand, or they can adapt by engaging in reversible cell injury, or they can die, which is when there's irreversible cell injury. So when there's an increase in demand or reversible cell injury, if the stress is removed, the cell goes back to its original state. If there's persistent stress, the adaptation continues, and you have the mechanisms of cellular adaptation called hypertrophy, hyperplasia, atrophy, metaplasia, dysplasia. And then there's also some other responses the cell can have, such as storing materials within the cell. If the stresses are relieved, the cell goes back to normal. If the stresses are not relieved, then you have irreversible cell injury and eventually cell death. And we're going to talk more about all this stuff. So on to objective 2, explore concepts in cellular adaptation. So cellular adaptation, the mechanisms as outlined before are atrophy, which is a decrease in cellular size. So you notice the prefix A, that means not against, and troph means grow, so it's not growing, it's getting smaller, in other words. Hypertrophy, the prefix is hyper, that means bigger, so more growth. Hyperplasia, which is increase in number of cells, so that is where the reproductive rate comes into the picture. And dysplasia, which is a change in cell shape, size, but still can be reversible, still an adaptive change. And metaplasia, which is where one adult cell type is replaced by another. And that is usually caused by chronic stress. So An example of hypertrophy would be what happens during the menstrual cycle when this endometrium gets larger, and then there's hypertrophy, and when the lining sloughs off. Hyperplasia, an example of that is a callus. Dysplasia is an example of dysplasia is disorganized cell growth, and metaplasia, an example of that is like when a person is a heavy smoker and the bronchial lining changes from one type of cell to another type of cell, which is less functional. So let's talk more about this. So atrophy is cell shrinkage. It can be caused by denervation, which is where there's a decrease in nerve supply, nutrient starvation, chronic inflammation, hormonal stimulus. And the mechanism is that, and also hypoxia can cause this, is decreased protein synthesis, increased catabolism, and sometimes autophagy is also part of it. So there is a normal cell and then a squunched down atrophic cell. Hypertrophy is increased cell size. It's caused by increased demand. It's a normal response to increased demand, stretch, and pressure. So, good example, muscle cells, striated muscle cells responding to exercise. And in this case, there's an increase in cell protein and growth factors are triggered. So in hypertrophy, individual cells become larger. Hyperplasia is an increase in cell number. That is usually stimulated by stress severe enough to cause some cell death. And that is, the mechanism is increased mitotic cell division. So there you see more cells, but they're the same type. The cell type doesn't change. Metaplasia is the replacement of one cell type with another. A stimulus for this is persistent cell injury. The mechanism is the reprogramming of precursor cells. So an example is as I mentioned before, often happens in glandular epithelial cells when they change to squamous, or bronchial epithelium changes to from ciliated to columnar. So those are two examples. And that's a picture of that. And there's the smoking man. So dysplasia is abnormal cell size, shape, or organization. The stimulus is, again, repetitive stress or infection. And it's designated as a low or high grade, depending on the extent. So let's talk about the causes and effects of cell injury. So those were adaptive cell changes. And now we're going to look at when the stress is not relieved and the cell is actually damaged. So here, this slide just shows you some pictures of depicts the different causes, some of the different causes of cell stress. So there's intrinsic type, or within the cell types of stimuli, and those would be metabolic, degenerative, neoplastic, immune, or nutritional. And then there's external or environmental stresses, which are listed here. And so those are the stressful responses. And down at the bottom, it shows some cell changes that happen in non-stressful conditions. So here's kind of the big picture. So there's a normal cell, there's stress, the cell adapts to the stress, and in that case, their homeostasis is maintained. But if the stress is persistent, then then there's irreversible cell injury and the cells die. So drawn reserves, adapt or die. So here are some things that cause cell injury. The hypoxia is the most common. Chemical injury, that would be like from heavy metals, lead, or drugs like therapeutic drugs taken or non-therapeutic drugs taken. Temperature, which is hot or cold stress, frostbite or burns. Nutritional deficiencies, which just deprives the cells of the materials they need to work. Injuries, intentional or unintentional, and free radicals and reactive oxygen species, which are byproducts of oxidation. As you may remember, oxidative phosphorylation and how it forms ATP. So reactive oxygen species are a byproduct of that. However, hypoxia is the most common. So there's ischemic injury from that, anoxic or reperfusion injury, 3 different types. So what happens when you have cell injury? So the cell responds with a decrease in ATP, causing failure of the sodium pump and sodium calcium exchange, which means that the cell can't control its environment and it can't pump water out of the cell. So then you get a response called hydropic degeneration or swelling. And also cell water can be isolated in vacuoles, so that's vacuolization. So ischemic and reperfusion injuries are another type of injury. So ischemic injuries are caused by lack of oxygen, which brings about these cell changes. And then, unfortunately, it was discovered that more damage happens once the blood supply is re-established. This was discovered with the advent of things like streptokinase, the clot busters. When blood flow was restored to heart muscle and also brain tissue, there were reperfusion injuries that occurred. And this is because when a cell undergoes ischemic injury, there's decreased oxygen, which turns off the pumps, as we talked about. And then when the oxygen level is restored, there's an increase in fluid movement in tissues, an increase in metabolism, which increases the release of reactive oxygen species, and that triggers inflammation, and that causes tissue damage. So oxidative stress is one mechanism. Increased intracellular calcium is a consequence of a lack of pumps working, which then leads to inflammation, and then that triggers complement, which is an inflammatory response, which we will talk about more next week. So this is what it looks like. You have the oxygen not getting through the blood vessel, and so then you get the swollen cell. And then when the oxygen is moving again, you get the reactive oxygen species, peroxide, and radicals, and you end up with a necrotic cell. So what, you ask, are free radicals? Now this is my little idea of a joke. Free radicals always make me think of the 1960s and 70s, so haha. Free radicals and reactive oxygen species are electrically uncharged atom or group of atoms having an unpaired electron that damage lipid peroxidation, alteration of proteins, alteration of DNA, and mitochondria. These reactive oxygen species are called that because they easily react with other molecules. and have ill effects on DNA and RNA. So this is just an image that shows the actions of reactive oxygen species. So the peroxisome is important because that's one way that the cell maintains balance between the beneficial and deleterious effects of reactive oxygen species and free radicals. That's mainly the gist of this slide. Explain reversible cell injury and intracellular accumulations. This is objective 4. We're plodding away here. So this is another type of cell injury mechanism. So it happens when ATP is depleted, resulting in mitochondrial damage. Then there's accumulation of oxygen and oxygen-derived free radicals, causing membrane damage, which then results in protein folding defects and increased intracellular calcium concentration and loss of so calcium builds up. And then this results in these two consequences: hydropic cell swelling and infiltrations and accumulations. So hydropic cell swelling looks like this. You can see the big cell and the little cell, the normal cell. It's usually the first response to stress. It's usually due to a loss of cellular pump function because of hypoxia, and it results in the cell not being able to move water out of the cell, because when there's no oxygen, there's no pump. When there's no pump, the water doesn't move. So cell swelling, this is an image that depicts cell swelling and hydropic degeneration. So there's an injury, sodium, water move into the cell, potassium moves out of the cell, and then there's all these vacuoles that are formed, and there's swelling of the cytoplasm, and that's what that looks like. So another way that this can be manifested is our cellular inclusions or accumulations. Cellular accumulations are also known as infiltrations, and that's just extra stuff that's kind of walled off in the cell, and things like water, lipids and carbohydrates, glycogen and proteins are normal materials that can be in inclusions or accumulate. But then you can also have abnormal materials that accumulate in the cell, such as pigments or abnormally folded proteins or metabolites. And then there's non-degradable compounds like pigments and other things like silica. So like, for example, if a person has jaundice, that you can see that because there's an accumulation of the pigment that's a byproduct of bilirubin in the cell, and or a person that's exposed to coal dust will have silica, which is just a non-degradable compound that's stuck in the cell and interferes with its function. And the bruising is another example of an inclusion. And that's when you see the hemosiderin and the iron-free pigments that build up in the cells, but it's temporary. So I ask you, which of the following is the most common cause of cellular injury? Which answer do you think? Describe irreversible cell injury and cell death. So there's, as we discussed, there's two types of cell injury. There's reversible, and then there's non-reversible. So another way of describing this is sublethal, which is reversible, excuse me, and lethal, which is not. So the difference between the two is the duration of the stress and whether the stress is relieved. So types of irreversible cell injury include autophagy, apoptosis, and necrosis. So this is kind of the process that happens. So you have the normal cell, you may have reversible cell injury, which is transient. If the stress persists, you get adaptation. If it is unable to adapt, then you get cell injury. If the stress remains severe and progressive, you get irreversible injury. Then you can either have cell death from apoptosis or necrosis. And we'll talk about the difference. So autophagy is a mechanism for selective, so this is a physiologic mechanism quite often. It is a mechanism for selective removal of unneeded proteins and organelles. So it's really a self-destructive and a survival mechanism. So when unneeded compounds are tagged for recycling by ubiquitin, then the ubiquitins attached to it. The tagged compounds are then taken to proteasomes, which is like a garbage can, and recycled. So this autophagy plays a major role in the aging process. I think that's super interesting that process happens. So this image just shows a misfolded protein that's being dumped. So apoptosis is another process which is also physiologic, and it's kind of like shedding of cells. It's triggered intracellular suicide. It's tidy and it's non-inflammatory. It is programmed cell death. So usually it's stimulated by survival signals or self-monitoring. It is, the mechanism is that blebs are formed and vesicles, and then that attracts phagocytes, which get rid of the cell. It's physiologic. So physiologic applications of apoptosis are bone remodeling, sloughing of the uterine lining, and in fetal development, there's lots of, as the fetus goes through different stages, some cells disappear and others are formed. And then there's pathologic, for example, autoimmune diseases are an example of that. So this image shows the process of apoptosis. So what happens is the cytochrome C leaks into the cell, which triggers capsases, which are enzymes within the cell. Then that sets off a cascade. The cell then shrinks, rounds up, and then forms blebs, which attract a phagocyte. And then the cell fragments, and then parts of the cell organelle can be reused. And what's nice about it is that there's no triggering of the inflammatory response, so there's no surrounding cell damage. On the other hand, we have necrosis, where there is surrounding cell damage. That is cell death due to injury. What happens here is the ATP-driven pumps are stopped. That allows calcium to rush into the cell. The mechanism is that mitochondria, nuclear, and plasma membranes dissolve at that point. So this is an image that shows that process. So necrosis is the sum of cellular changes after local death and the process of autodigestion, which is also known as autolysis. So there's different types of necrosis. There's coagulative, liquefactive, caseous, fat, gangrenous, and then a subcategory of gangrenous is gas gangrene. So this is what coagulative necrosis looks like. It's most common in kidneys, the heart, and adrenal glands. And the necrotic material is protein denaturation. Liquefactive necrosis is more common in brain tissue. It's often caused by abscesses or infection. And that is triggered, or what causes the cell breakdown are hydrolytic enzymes. And quite often, as I say, bacterial infection is the cause. So that's what that looks like. Caseous necrosis, caseous means cheese-like in Latin. And it is a a combination of coagulative and liquefactive necrosis. And what happens is like for example, in tuberculosis, there is the infection is walled off in a thing called a tubercle. And then the material so then the macrophages, or the infection fighting cells that are within the tubercle, digest all the stuff that's in there, but then it's trapped in there. It often becomes what's called a granuloma, which you'll hear more about later. Then there's fat necrosis, which is injury to destruction of fat cells, more common in breast, pancreas, and other abdominal organs. So what happens in this case is lipases destroy the fat cells, and then the oily cell contents replace the cells, and you get loss of functional cells there. That is commonly seen in liver disease. Gangrenous necrosis is a type of ischemic injury. It's death of tissue from severe hypoxic injury. So the dry gangrene is typically on extremities, and it's usually self-contained. So you'll have like a black toe you might see, and the tissue surrounding it is healthy. Usually the extremities cool and pale because there's not very good circulation. Excuse me. But it's localized. Whereas wet gangrene is when there's the infection invades the deeper tissues, and then it's no longer walled off. And then finally and so then there's an active goopy infection that happens. And then there's gas gangrene, which is usually caused by Clostridium. And the bacteria release toxins and this foul-smelling gas. Usually you can tell a person has it because they have bubbles under the skin, and then there's blisters that have this smelly exudate. So here are some internal organs that are necrotic, and those are some different terrible things that can happen. Volvulus, thrombosis, strangulated hernia, so intussusception. It's a little more common in children, but not exclusively, and gangrene. So I ask you, which type of necrosis best describes the death of a cell from hypoxia, generally as a result of ischemia in the lower extremities? Fat, coagulative, liquefactive, or gangrenous? So let's talk a little bit about cell aging. So cellular aging is the progressive decline in proliferation and reparative capacity of a cell. It can be caused by exposure to environmental insults, external forces, or it can be caused by intrinsic forces like DNA, broken telomeres, you may recall what those are, accumulated products of metabolism, such as free radicals. And the features of cell aging are hypertrophy, which is a thickening and increasing in bulk. Hyperplasia is another feature. So the mechanisms of cell aging are protein clumping, autophagy, telomere shortening. So every time there's a reproductive cycle, the telomeres get a little more brittle. That's the ends of the chromosomes. Disruption in cell signaling pathways, which control gene transcription. Decrease in nuclear trafficking of mRNA. Damage of proteins and porous leaking of nuclear membranes. So the reproductive process is messed up, resulting in misfolded proteins. Calling of damage or misformed proteins decreases. So you get a buildup of all these misfolded proteins within the cell. For an example of that is unfolded proteins, which lead to Parkinsonism and dementia. Dysregulation of autophagy of the organelle, so things don't get removed like they should be, or it happens more than it should. mutation of mitochondria, cytoskeletal disruptions from oxidative stress, which then triggers more apoptosis, and changes in the cytoskeleton, and plasma membrane, which disrupt longevity signaling pathways. So there actually are pathways that there's signaling pathways which tell the cell to continue to live, and those get messed up. So, that is not a very good picture, but that is an image of some of the things that happen with cellular aging that we just talked about. Aging, physiological aging or tissue aging, there are different terms for this. There's cellular aging, which we have been talking about, which is the physiologic deterioration over time. And that the mechanisms include atrophy, decreased function, and loss of cells, and a gradual loss of homeostatic mechanisms, usually unknown cause. Cell senescence is a natural culling of unwanted cells. So that happens throughout life. That's a natural process. On the tissue level, there's tissue changes that happen are progressive stiffness and rigidity as there's more inelasticity in the collagen. So there's increased rigidity of collagen, decreased muscle mass, loss of height, pelvic widening because the bones are getting softer and the tissues holding the bones together are less vigorous. Blood vessels become more rigid. There's a decrease in bowel motility, decrease in muscle tissue and fat, and a loss of lung compliance. These are all normal changes of aging. And so another term that is used to describe this process is frailty, and that's a clinical syndrome in which there's unintentional weight loss, which that would be reflected in loss of muscle mass, self-reported exhaustion because of loss of muscle, and weakness, slow walking speed, and low physical activity. Which statement is true about cellular change due to aging? And I'll let you read that and figure out the answer. So let's talk about tissue death. So on the body level, that's what somatic means. That is the death of an entire person. There's no inflammation or immunological changes prior to death, so it's a natural process. That's when a person dies, from a disease process or something. There's a cessation of heartbeat, respiration, and brain function. And then there's post-mortem changes that happen. Algor mortis, which is body cooling. Liver mortis, which is when the fluids drain to the lowest point, and there's sort of a purpley color to the tissues and the lower parts of the body. Rigor mortis is when the actin-myosin connections are unable to detach. That results in rigidity. And then finally, there's post-mortem autolysis, when the enzymes are released from the cells and the tissues deteriorate. So the signs of somatic death include no pulse or respiration, and then no ECG, which reflects brain function. The body is no longer able to regulate temperature, which is called poikilothermia. And then after clinical or somatic deaths, the cells continue to live for a while, and they die at variable rates. So you see some of these changes, body cooling, changes in the eye, for example, early signs of somatic death include pallor, cooling, dilated pupils. As the fluid drains, you get these peaked features, and then you will see lividity. That's when all the blood drains. And then cellular death begins to occur at 6 to 24 hours, and that's when you see rigor mortis. And that's when the muscles start to protein seize up. And then finally, you will see 24 to 48 hours after death putrefaction, and that's when the tissue becomes greenish, there's bloating, and the skin starts to slip off if you touch it. So that, on that cheery note, that is the end of my lecture, and thank you for listening.